Hello friends, good evening all. I am Dr. Koles. Welcome you all to this lecture. I am a veterinary assistant surgeon working under Government of Odisha. Today the topic of discussion is brucellosis, the most fatal as well as the dangerous disease when production is concerned in livestock sector. So let's discuss what is this disease. This disease also known as Mediterranean fever or Andolan fever or Malta fever. As this disease is profoundly found in Mediterranean region, so the disease is named so. Brucella is a zoonotic and infectious disease, means it transmitted from the animals to the human beings, and it is due to the or it is caused by gram-negative Coccobacillary bacteria, and this belongs to the genus Brucella. It is primarily an infectious disease of cattle and animals, domestic animals, characterized by the very important abortion in the late gestation and formation of the granulomatous lesion in the genital organ, joints, and fetus. So, three characteristics it generally exhibits: abortion in the late gestation. First, second is the formation of granulomatous regions in the genital organs, like in vagina, like in penis. So, this forms the granulomatous region, as well as the swelling of their joints, as well as the liver and the peculiar types of lesions found in their liver of the growing fetus. So this disease is now commonly found in case of developing nations, because already the developed nations have already eradicated their this disease through their effective control measures. So, coming to the history, this disease is now called as brucellosis under the name the Mediterranean fever. First, when there was a Crimean War was there in the year around 1887. So, this disease is named so, and the disease is first established by Dr. David Bruce. In 1987, the Danish veterinarian Bernhard Bank isolated the Brucella abortus. Very important, the scientist Bernard Brand, as he isolated this organism, so the name is given so, the Banks disease. Maltese doctor and archaeologist Dr. Tenny Jamit identified unpasteurized milk as the major source of the pathogen in 1905, and it is has since has become the Malta fever, because the Malta is named as the doctor is from the Maltese. And in cattle, the disease is known as contagious abortion or infectious abortion, as this disease causes abortion in the last trimester of the pregnancy, means third trimester, means last stage of your pregnancy. The popular name is ondulant fever, originated from the characteristic ondulance or wave-like nature in the fever, which rises and falls. Means first there will rise in fever, then fall, then again rise in fever, then fall. So it is just the wave-like structure. That's why its name is an ondulant fever. So generally, the many livestock are greatly affected with this disease. In goats and sheep, the Brucella melitensis, it is a frequently asked question in JRF examination. Brucella suis in pig, Brucella abortus in cattle, Brucella obese in sheep, and Brucella canis in case of dog. In this lecture, I will only deal with the pathological aspects of the disease. In next slide or in coming slide, I will give you the detail structure, the general, the microbiological structure, the medicinal aspects, the preventive measures, all these things I will tell. But in this lecture, I will only deal with the pathological aspects. So there are many modes of transmissions are possible. First of all, the ingestion through your ingestion means through taking of contaminated feed or water, penetration of intact skin or abraded skin. It can also penetrate into the body through artificial inseminations of the infected semen. Very important, the infected bulls which previously affected with the Brucella organisms, the cow might get the infection and the contamination of the udder during milking, as the milk always present in the unpasteurized milk, so the contamination of the milking during milking also a possibility. And some literature is telling flea rides, dogs, and other contaminated equipment like the fomites are also a major source for infections. So coming to the pathogenesis, the main first of all these organisms localize in lymph nodes and spread via lymph nodes to the spleen and mammary gland, and subsequently the organism resides at the reproductive organ, placenta, and the fetus as well as the udder. Okay, the final target organ is your reproductive organ like your vagina, penis, cervix, placenta, where the fetus generally grows, the growing fetus as well as the udder. Why this organisms occurs in the reproductive organs like in fetus, placenta? Because the placenta secretes a certain kinds of substance that favors the growth of this organism, that is known as your erythritol. Very important question. This is erythritol. This substance is secreted by the placenta, favors the growth of this organism, as well as when this organism is phagocytosed by the your phagocytes or lysosome, or phagolysosome better to say, this Brucella organism releases this enzyme 5 des guanine and adenine, which are capable of inhibiting the degranulation. Means it cannot make, or it cannot have a impact for itself for degranulations of peroxidase containing granules. That's why it inhibits the myeloperoxidase peroxide halide system of the bacterial killing. So this is a very complex system of bacterial killing. I don't know actually what the exact mechanism is. Myeloperoxidase peroxide halide system of bacterial killing. These systems cannot break down these bacterial organisms. So as this is a intracellular organisms, means this resides inside the cells. The bacteria resolves the granuloma formations in the RES system, reticuloendothelial systems, and this leads to the hypersensitivity reaction. So mostly, as I have told already, this disease is manifested as by the symptoms of abortion in the last gestation around 7 to 9 months, and the retention of placenta with and metritis, inflammation of your uterus. Better to say endometrium. In the orchitis, the inflammation of your scrotal sacs, and as well as the epididymis in bulls leading to the sterility. So hygroma of the, I've already told, in joints also there is formation of the granuloma. So this is the conditions. This is the hygroma of the knee. During this slide, I want to give my thanks to the Sushan Panda sir, because I have already taken his slides. Actually, I want to disseminate the knowledge to those people, those who have don't have any idea regarding the brucellosis, or have or want to upgrade the existing knowledge, or the refresh the existing knowledge of the vets. Those people who have already detached from the academics since a long time. So coming to the gross lesion, what we will find the edema of the fetal membrane and the leathery texture. When we go for the your palpation through the rectal route, we will find a leather-like structure. So this confirms of your presence of brucellosis organisms. Obviously, when the organism is present inside your cotyledons or in the placenta, they will find the necrotic cotyledons, granulomatous lesions, and focal necrosis in the several organ. It also, in very many instances, we have found the possibility of the bronchopneumonia, as well interstitial pneumonia, and as this organism present in the order, it causes the induration of the mammary gland in case of cows. So histological findings, we will find the granuloma and necrosis of liver. We find this is, and the phagocytic cells, epithelial cells, and lymphocytes surrounded by the proliferative tissue. Bronchopneumonia organisms is discombinated in epithelial cells of chorion. That's fine. Then epididymis leads to fibrosis, and it causes a permanent sterility in case of bulls. So we have to first go for your diagnosis. This is very much important, and brucella ring test to detect the antibodies in case of milk. So brucella ring test. So there is also a specific reagent. We go for the sampling of your. Generally, we do the testing of a milk samples through brucella ring test, and through ELISA and the CFT complement fixation test, and the fluorescent antibody test, FAT, we can diagnose the presence of this organism. And another is your Rose Bengal precipitation test, RBPT. Generally, in our department or in our state, generally this test holds a very significance in identifying this organism. So this, and coming to the, so this much is in this lecture, and in the next lecture we will deal how to